Hey everybody, so in this video we are going to take a look at an important concept in genetics, and let me introduce the concept by diagramming two genes, A and B, like I normally do. And between A and B there, we are going to, let me see, am I recording? Yes, I am. So between A and B there, let's say the distance is 25 centimorgans. Okay, so we know from the last video that in an individual that is doubly heterozygous for those two genes, linked genes, I should say, that, we should be able to predict the number of gametes with distinct phenotypes produced by distinct genotypes produced by this individual right here. So these are the recombinant gametes and 25 centimorgans, so 25% should be here, should be those, and 75% should be this one. But what happens, if B was way out here, a little further away? Well, then we would predict, which is most often the case, that, okay, this goes up, let's say 37.5 centimorgans. So we'd have 37.5% recombinant gametes and 62.5% non-recombinant. What if we go all the way out here? Do we ever go up to 75 centimorgans? Is that possible? Is it possible? Well, in some ways it's possible, and I can explain that in a moment. But is it possible, if these are 75 centimorgans apart, for us to have 75% recombinant gametes and 25% non-recombinant gametes? Is this possible? And in this video, we are going to learn that it is not possible. It is not possible. The theoretical limit to the number of recombinant gametes is 50%. So even if B is located more than 50 centimorgans from A, the maximum number of recombinant gametes we can get is 50%. And we're going to see why that is the case with this little exercise. And you should probably check out the notes. I have a little, at the end of the notes, I have a worksheet that you can do to help you see this. Before we go over the worksheet, I want to give you this formula. And what is the formula? So if you can take the percent of recombinant gametes, so if you know the percent of recombinant gametes, and you multiply that by 2, you can know the percent of tetrads that crossed over between the genes that you were interested in. So let me see, a better way to say that, percent recombinant gametes times 2 equals the percent of crossover tetrads. And we can have another formula based on that one, which is the percent recombinant gametes is equal to the percentage of crossover tetrads divided by 2. Okay, so if you know the percent of crossover tetrads, you divide that by 2, you can figure out the percent of recombinant gametes. So these formulas are useful, but let's see where they come from. So let's see, I tried to zoom in here. And I hope this works out. You know, as I was taking notes before, let me see one thing. I don't know I was mostly on camera for those last 2 pages of notes. So let's move forward, plow forward through this worksheet. Let me sharpen my pencil again. So now on this worksheet, and again, this is in the notes, and you can go through it on your own. You should go through it on your own, but I'll explain what's going on here. So what what I'm showing you here is in each row here, we have 5 rows. I am showing 5 different cells undergoing meiosis 1. So 5 different cells. And in only one of these meiotic cells is there a crossover between genes A up top and B down bottom. And what is the genotype here? The genotype is A, B, little a, little b, for all of these. So again, you might need to pull up the notes so you can see more clearly what these labels are. And what I'm showing you here is that only one of the 5 cells, only one of these is undergoing a crossover. So 1 out of 5 is 20%. So we have 20% crossover tetrads. And I spelled it wrong there in the example. So 20% of the tetrads pictured here are undergoing the crossover between A and B. So if we use that formula, divide that by 2, we should see 10% crossover gametes. And here are the crossover gametes right here. 1, 2. So if you wanted to count these up, 1, 2, 3, 4, 8, 12, 16, 20. We have 20 gametes in total. Only 2 of them are undergoing the crossover. And again, in the notes, these are colored so you can clearly see the 2 is only 2 out of 20 are crossover gametes. So 2 out of 20 is 10%. Okay, everything makes sense. So let's go down here. 2 of the tetrads are undergoing a crossover. 3 are not. So 2 out of 5 is 40%. So 40% crossover tetrads. Divide that by 2, we should see 20% crossover gametes. And again, if you have the color version, 1, 2, 3, 4, you see 4 out of 20 are crossover gametes, so that's 20%. So down here, 60% crossover tetrads. If you divide that by 2, according to our formula, we should see 30% crossover gametes. So let's count them up. 1, 2, 3, 4, 5, 6. Okay, 6 out of 20 crossover gametes, that is 30%. 30% CO gametes. Okay, I think you might see where we're going by now, right? Okay, so here we have 5 meiotic cells. 4 of them have tetrads that are undergoing a crossover between A and B. That means 80% of the tetrads are crossover tetrads. That is going to give us 40% crossover gametes. And we can double check. 1, 2, 3, 4, 5, 6, 7, 8. So 8 out of 20, that is 40%. Crossover gametes. And finally, down here, we have 1, 2, 3, 4, 5. All of the meiotic cells are undergoing a crossover event. Well, the tetrads in each meiotic cell are undergoing all of them are undergoing a crossover event between A and B. So 100% crossover tetrads. They produce 50% crossover gametes. And we can count that up, 10 out of 10. So, yep, okay. Oh yeah, this is where I was supposed to be counting the numbers, right? So 10 non-crossover, 10 crossover gametes. Okay, and here, what do we have? 4 crossover, 16 non-crossover, and you can go through and fill out the numbers if you want. But the point is, even if we have 100% of the tetrads undergoing a crossover between genes A and gene B, we only get 50% crossover gametes. So even if all of the tetrads experience a crossover event, only at most half of the gametes can be crossover gametes. So that's why the theoretical limit TL of crossover gametes, COG, is 50%. And you can kind of see why that's the case, right? So even with this tetrad right here that's undergoing the crossover, 2 of the gametes are crossover gametes, but 2 of them are non-crossovers. Okay, so that's why we have this theoretical limit to 50% to crossover gametes. But that leads to a question. How can we have something if these genes are 25 centimorgans apart, and these ones are 35 centimorgans apart, say right here, 25 centimorgans between A and B, and 35 centimorgans between B and C. Won't these be 60 centimorgans apart? And yes, so they are 60 centimorgans apart. But in order to actually determine these distances with genetics, well, you would need to do these crosses separately and show that, okay, yeah, A is 25 centimorgans from B, and B is 35 centimorgans from C. So we wouldn't be able to measure this distance accurately by doing a genetic cross, let's say, by doing something like this. Because the theoretical limit to crossover gametes is 50%. So we wouldn't be able to get an accurate distance between these two. And we'll see more on this in the next lecture. Okay, so that's it for lecture 7. Let me know if you have any questions. I hope you were in lecture, and if you had any questions during lecture, I hope these videos help answer some questions. And if not, just email me, or come see me in office hours, or make an appointment for extra office hours. Okay, thanks. See you on Thursday.